In this short video, we will examine the genomic neighborhood surrounding the target gene, Rheb, in Drosophila melanogaster. Navigate to the GEP UCSC Genome Browser Gateway page. Click on D-melanogaster in the UCSC Species Tree and Connected Assembly Hubs table. Ensure the August 2014 assembly under the D-melanogaster assembly field is selected. Enter Rheb in the Position Search Term field. Click on the Go button. On the following fly-based protein coding genes page, click on Rheb-RA at chromosome 3R 5,568,921 to 5,570,491. Since both Rheb-RA and Rheb-RB span the same coordinates, it doesn't matter which of these two we select. FYI, if the coordinates weren't identical, we would have chosen the one that covers the largest region for this step. Because the Genome Browser remembers our previous track display settings, click on Default Tracks in the Display Controls below the Genome Browser image. The Default Tracks button quickly removes any display changes you previously made and returns to the defaults. There are three major sections of the Genome Browser: a set of navigation controls, the Genome Browser image, and a set of track display controls. In the fly-based protein coding genes track, we should now see the gene structure for the two isoforms of the Rheb gene, Rheb-RA and Rheb-RB. Notice that Rheb is on the chromosome 3R scaffold. Isoforms are potentially different versions of a protein encoded by a single gene. Isoforms result from alternative splicing of a particular pre-mRNA and/or the use of a different transcription start site. In this example, proteins A, B, and C are isoforms encoded from the same gene through alternative splicing. Zoom out until you can see the nearest two genes on each side of Rheb. We are now viewing the genomic neighborhood of the Rheb gene in D-melanogaster, the region of the scaffold containing Rheb, the target gene, and its neighboring two closest upstream genes and two closest downstream genes. The genomic neighborhood of Rheb includes CG12746, CG2931, CRIMP, and CG2926. FYI, each protein coding gene annotated by flybase in D-melanogaster has an annotation symbol that begins with the prefix CG for computed gene. Unless genes are characterized experimentally and formally named, they are referred to by the symbol. For example, in the figure above, 3 of the 5 genes, CG2931, CG12746, and CG2926, have not been named since they haven't yet been characterized experimentally. However, the Ras homolog enriched in brain and collapse and response mediator protein genes have been characterized by past scientific studies, and thus are referred to by their gene symbols Rheb and CRIMP respectively. Now we need to draw a sketch of the genomic neighborhood of Rheb in D-melanogaster. To do so, we must identify the direction of transcription for each gene by zooming in on an intron or exon of each gene. Start by zooming in on an intron or exon of Rheb. Since the arrows within the introns of Rheb point to the right, in our sketch we need to draw an arrow pointing to the right and label it Rheb. Repeat these steps for the two closest genes on both sides of Rheb. The arrows within the introns of Rheb point to the right. Therefore, we know Rheb is transcribed from left to right and located on the positive strand of DNA in D-melanogaster. Since Rheb is our target gene, the gene that we are annotating, the two closest genes on its 5' side are considered upstream, and the two closest genes on its 3' side are considered downstream. The two closest upstream genes of Rheb in D-melanogaster are CG2931 and CG12746. The two closest downstream genes to Rheb in D-melanogaster are CRIMP and CG2926. Since the direction of the arrows within their introns point to the right, Rheb, CG12746, and crimp are on the positive strand. In contrast, CG2931 and CG2926 are on the negative strand since the direction of the arrows within their introns point to the left. FYI, as we learned in Understanding Eukaryotic Genes, Module 1, genes on the same DNA molecule may be transcribed in opposite directions. Genes on the positive strand of DNA are transcribed from left to right, and genes on the negative strand of DNA are transcribed from right to left. Since genes have directionality, the areas of DNA we call upstream and downstream change depending on the orientation of the target gene. Since river streams have directionality, they flow in only one direction. To help you orient yourself, think of your target gene as a raft floating down a stream. Wherever the raft just floated from is upstream of its current location, and wherever the raft is floating to is downstream of its current location. Notice how the areas of DNA we call upstream and downstream to the target gene change depending upon whether the target gene is on the positive or negative strand of DNA.